Okay, we now start with the second topic of the 8th chapter, and this is on animal welfare indicators. And you already had a practical week in which you could practice the scoring of some welfare indicators that were relevant for poultry welfare. But I will talk about animal welfare indicators more on a theoretical basis. Before we can decide which indicators we want to use if we want to assess animal welfare, we need to have more information about the context and the goal of the welfare assessment that we want to do. Questions that we need to ask are, for example, do we want to assess the welfare of this individual or of a certain social group, or the welfare of all the animals on a farm or a region or a country, and so on, because these will have a big influence, of course, on which are the good indicators you need to use. Another thing we need to have clear is, do we want an animal welfare assessment at this moment in time? How does the animal feel right now? Or do we want a historical assessment? How was the welfare of this animal during the last year, from now until a year ago? Or do we also want to include what are the risks for the welfare of this animal? And then it becomes more also focus on the future, a sort of risk analysis for the future. So we have to make it clear also which stage of the production we want to do an animal assessment for. For example, in the welfare quality protocol, they state clearly that for the broiler chicken protocol, you go from the rearing, the producing, and the end of life stage. Whereas the laying hen protocol does not include the rearing phase, also not the end of life phase, but only the period during which they are producing eggs. So it's very important to be very explicit about the context and the goals of your welfare assessment. Another thing you need to be very explicit about is the application. What do you want to do with it? Why are you doing your assessment? Do you want to do an exhaustive welfare assessment? Or do you want to focus on the most pertinent issues, the biggest welfare issues for that population or that group of animals? Or you want to have an as elaborate assessment as possible? Is the idea to do monitoring, where, for example, it is important or it might be important, so do you have an assessment that you compare a historic assessment with an assessment later on in the date? Do you want to be able to compare systems, countries, companies? What's the purpose of the assessment? Is the purpose for a labeling scheme or a certification scheme, or to benchmark different farms, to give feedback to farmers on how they can improve welfare on the farm? Or is it for research purposes? So these will all have a very big impact on what would be a suitable indicator to use. What are general features of a good welfare indicator? What are the criteria for a good indicator of welfare? First of all, it ought to be a valid indicator, which means that it really tells you something about the welfare of the animal, or at least about one of the aspects of its welfare. For example, you could have a measure of welfare that estimates the prevalence of animals showing stereotypic behavior. For me, this is a less valid measure of welfare than the incidence of stereotypic behavior. And the number or the percentage of animals starting to show stereotypic behavior over a time period. Because it's the development of stereotypic behavior that is more relevant than animals showing stereotypic behavior. Linked with that validity is that we would prefer to have an indicator of welfare that is animal-based instead of the description of the management of the housing system, because, as argued before, these are rather risk factors rather than direct indicators of welfare. And then it has to give information about the correct time frame, and that links with our objective. Do we want to do a snapshot evaluation and a historical evaluation, also future risk analysis? So the indicators we can use, for example, I will use an indicator on lesions. We could say we do an assessment of aggressive behavior, and we do an observation on a farm, and we see how much aggressive behavior there is. And this is a very instantaneous assessment. For that gives information about the present state. You could say most of the time we're not interested in that. We want more kind of an assessment that is valid for a prolonged period of time. So we are interested in a bigger time scale than this. And then you could say, for example, okay, we're not going to look at aggressive behavior, but we're going to look at the effect of aggressive behavior on the skin and count and quantify the skin lesions of the animals. You could say, well, we want this time frame because we will only go to a farm 2 times a year, so we want this time frame to be as long as possible. And then it could be an argument to also include old skin lesions and not only count the fresh skin lesions. So you have to be very clear about your time frame. A very important aspect for a good welfare indicator is also that the indicator is reliable. And that concerns the inter-observer reliability, the intra-observer reliability, the test-retest reliability, not being subjected to expectation bias, so you have an objective measurement. If you need to assess a sample of animals out of a flock, that your sample is representative for the entire flock. So these are all issues that relate to reliability. And very often it is also important and a challenge to have an indicator that is logistically feasible, that you have enough time, resources to do that it doesn't impose a hygiene risk to the farmer, that it doesn't disturb the animals too much, and so on. And very important also, especially if we want to do, if the purpose of the assessment is to be able to compare the evolution in time or to compare farms, is that your indicator is robust and not moment dependent, so that you have a high test-retest reliability. That means that if I do an assessment on the Monday morning, I should have the same result as if I do the assessment on the Tuesday evening. So let's have a look at the welfare indicators that are used by the welfare quality project. So I focus also a lot on the welfare quality because it's still one of the most elaborate and most renowned protocols for farm animal welfare assessment. So if you want to find out more about the protocols and how they do welfare assessments using the welfare quality methodology, there's a link to the website where you can find a lot more information. What you see here on this big table is these 4 welfare principles that I talked about and the 12 welfare criteria. And you can see for the different species of farm animals the indicator that they use to give information about each of these 12 welfare criteria. So for example, for broiler chickens, they will look how many emaciated chickens there are in the flock. For laying hens, the second version of the protocol at least, they will look at the keelbone prominence. And so they try to have indicators for all of these 12 criteria. Although sometimes they have to acknowledge that there's no good criteria available, or it's not relevant to have an indicator for that criterion. You see that some of these indicators are in red, others are in green, and others are in blue. The red ones are not animal-based indicators, but are resource-based indicators. Welfare quality, they had this decision where they said that in preference they would use an animal-based indicator, but if there was no good or feasible animal-based indicator, they would use a resource-based indicator. So you see occasionally they use resource-based indicators and not animal-based indicators. You also see the green indicators. These are indicators that need to be derived from farm records or from talking to the farm. It's actually information the farmer needs to give you or that you have to retrieve from farm records. The reason I indicated that in green is because there could be a problem with reliability. It's one of these indicators for which we rarely have information about how reliable has that information been collected and can we really trust that. And then in blue doesn't come very often. In blue are indicators that are measured not on the farm but at the slaughterhouse. And for example, for broiler chickens, you see that rejections at the slaughterhouse is used as an indicator for absence of disease. So let's now turn to a couple of these indicators that are used by welfare quality. I won't cover them all, but I will focus on a couple of them because I find them quite interesting to discuss. Let's focus here on the indicator drinker space, which is used in the broiler chicken protocol by welfare quality to give information about the absence of prolonged thirst. So what does that indicator involve? Well, mainly look at how many drinkers there are compared to how many chickens there are. But of course you have different types of drinking systems, so you have to account for that. So the recommendation is for a nipple drinker there shouldn't be more than 10 chickens per nipple drinker. For cup drinkers there shouldn't be more than 28 chickens, and for bell drinkers there shouldn't be more than 100 chickens. And that's the indicator they use for assessing whether the animals are suffering from prolonged thirst. You can question, is this really a good indicator? Or can't we come up with better indicators? The first problem we need, or the first question we need to tackle, is it really needed to have an indicator of thirst for broiler chickens? One could argue, and it's an argument that is heard a lot by farmers and veterinarians, they say no, thirst is never a problem on broiler farms, because if the animals would be thirsty, it would mean that they would eat less and that would grow less efficiently. And therefore it would be economical suicide for a farmer not to prevent the animals being thirsty. And that makes sense. And yet you can argue, wouldn't there be a need for a good animal-based indicator of thirst? Could there be situations where broiler chickens, even on commercial farms, are sometimes suffering from thirst? And if we don't have a good animal-based indicator, how can we ever be certain? Because simply the number of drinkers per chicken can't be very accurate. We don't know how sensitive that is, and it doesn't persuade me as a good evidence that animals are not thirsty. For example, it can also depend on the flow of these drinkers, whether there are any defects, where they are being positioned, what the water quality is for these drinkers, and so on. But also it cannot be excluded that some individuals within the flock have difficulty reaching or operating the drinker. And then I'm thinking about, for example, these broilers that don't grow as fast as the other ones, as the average of the flock, those with a stunted growth, or those animals that are sick or weak or lame and therefore have difficulty accessing the drinking points. Also, most of these kind of nipple drinkers, they require the animals to drink from for them rather a natural position. And it's especially at high temperature when they have a need to pant, when this unnatural drinking position can make it difficult for them to drink. So especially at times when they need it most, the way that the drinkers provide water is not easy for them. You could ask, like, could there be good physiological indicators of thirst that can be used in broilers. And several of these have been suggested and investigated, and here you see a meta-analysis of the existing literature that has recently been published, which shows that indeed there are some of these physiological indicators that can be measured, such as osmolality, where typically it increases with the duration of water deprivation. So it seems to be linked with thirst. Same for vasotocin. Most publications seem to indicate that it increases with the duration of water deprivation, also with plasma sodium, and also, for example, the blood-packed cell volume. So you could argue these are kind of indicators that are animal-based that could be used as indicator of thirst. But of course, the problem with many of these indicators is that they're quite difficult to do, especially during monitoring. And it's not convenient to collect blood samples. And you could think, could there be easier ways of getting that information? And that doesn't require blood sampling. And then you could think about maybe the dry matter content of the excreta or of the intestinal contents. Could they be related to dehydration? But in a research by Van der Hassel, they found that these didn't correlate very well with the duration of dehydration. And they said these indicators listed here, they don't seem to have a lot of potential as animal-based indicator for thirst. And what would be easy to use would be indicators that doesn't require any lab analysis or any sampling at all, simply looking at a sample of animals that could give information whether they are dehydrated or not. And then indicators that are sometimes suggested include the capillary refill or the skin turgor or sunken eyes or the organ weights, but in this study they were not found to be well associated with the duration of dehydration. As illustrated in this graph, where you see the capillary refill time depending on for how long they have been dehydrated. So up to 48 hours. So even after 48 hours of dehydration, there was no statistically difference from animals that had not been subjected to a dehydration period. The only difference that was seen between some treatments was a kind of simulation of a chronic thirst, in which 10 days before slaughter, the broilers had access to a drinker for only 2 times 10 minutes each day. So it really meant more longitudinal possibility for thirst. So that was the only indicator or period when there was a significant difference with another value. What was found in this study, a little bit unexpected perhaps, was simply the blood volume was related to the dehydration duration. And so as soon as the animals were dehydrated for at least 36 hours, the volume of blood was significantly lower than for the non-dehydrated broilers. And that required, that is a measure that could perhaps be considered in a slaughterhouse environment, although you need to collect blood for 90 seconds. So it could be an indicator of severe thirst. And also the body weight. The more the animals were dehydrated, the less they weighed. So body weight could also be an indicator of severe thirst. But you could question, are these sufficiently specific indicators? Because body weight could be affected by so many other factors, and the same with blood volume. So what we really would need is a much more specific indicator of thirst. So you could think about designing a behavioral test, a very simple one. What will a thirsty animal do? It will try to drink. if it's available. And that could be the basis of a very simple test that was tested in this setup here, where animals were deprived for water for different durations, and then they were offered a new kind of drinker system they had never seen and experienced before, an open drinker space which is more natural to their drinking behavior. And we simply see how much water they consume over time. And as predicted, the group that was deprived for longest for water, they consumed water from this new drinker faster and more than the other treatments. It is important, of course, that that drinker is equally familiar or unfamiliar to all the groups you test, because they drink more from a drinker they're familiar with than from a drinker they're not familiar with. So we wanted to see that further, whether that could be a useful animal-based test of thirst. So we wanted to integrate this, what we call the spontaneous water intake test, into the welfare quality assessment. And we did this on 10 farms in Belgium and 10 farms in Brazil, where we wanted to test in the research what the validity and the sensitivity was under commercial conditions, whether there were unwanted confounding factors that could be the basis of a bias. And we wanted also to compare this spontaneous water intake test with the indicator that is used in the welfare quality protocol, which is drinker space. And the results were quite disturbing, because what we found, if we use the welfare quality test of thirst based on the drinker space, we would conclude that the highest score was given to the farms in Brazil. They seemed the least thirsty because there were relatively more drinkers per animal than was the case in the Belgian farms. However, when we did the spontaneous water consumption test with the open source drinker from which the animals could drink, and then see how much they were drank from it, much more was drunk on the Brazilian farms than on the Belgian farms. So if we use our spontaneous water consumption test, we would say the Belgian farms is having less of a problem with animals being thirsty, but if we would have used the welfare quality test, we would have come to the exactly opposite conclusion, that the Brazilian farms are the farms where the animals are less likely to be thirsty. We also saw that there were certain factors that have an influence on how much water is consumed during the spontaneous water consumption test. And these relate to the indoor climate and also to the stocking density. So if we ever want to use that test to compare farms over time or between farms, we need to somehow account for indoor climate and for stocking density. So what did we conclude from this research regarding the validity and the sensitivity of this spontaneous water consumption test? That it can detect 6-hour deprived chickens in Brazil, but in Belgium they had to be deprived for 12 hours for the test to detect that. As confounding factors, there was a country difference and a genotype, but we don't know which of the two is the most relevant ones, because in Brazil they mainly use the Cobb chicken, whereas in Belgium they mainly use the Ross chicken. But there is a suspicion that the country difference that we found was really a difference between genotypes, because it is known that the Cobb, they need to drink more for their body fluid homeostasis, and therefore may be more sensitive to dehydration. So if we want to interpret the outcome of the test, it's important to correct maybe for genotype and for barn climate. And we were very concerned that our test gave very opposite results from the welfare quality scores. There are also, both scores are also not correlated with one another. So personally, I have doubts about the validity of the drinker space test that is used in the welfare quality protocol. Let's turn to another example indicator. Let's stay with the broiler chicken. And now we go to the criterion of ease of movement. And for this criteria, they use the indicator stocking density, again a resource-based indicator. Again, we can ask whether the stocking density really gives sufficient information about the spatial needs of broilers. Couldn't we use animal-based indicators for that instead of a resource-based indicator? And there are some animal-based welfare indicators for broilers that are used a lot. That kind of a conventional set of indicators that are being used in broilers. And these include, for example, the tonic immobility as a test of fear, the latency to lie as a test of leg strength, the weight of the bursa compared to the body weight, the prevalence of hog burns, and the prevalence of foot pad dermatitis. And you can develop a welfare index based on these set of indicators. And if we do that, and we did this in an experimental setting in which we changed the stocking density from very low to very high, you can see that this integrated welfare score goes down as stocking density increases. And you can also see that in the beginning, you see an effect of leg strength and latency to lie. Later on, you see an effect on the hogs, and even further at very high densities, then you see a big effect on the foot pad dermatitis and even on the levels of fear as measured by the tonic immobility. But again, you can ask, are these rather the consequence of a high stocking density, or are these truly indicative of the animal's spatial needs not being fulfilled? And therefore, don't we need a more specific test of the spatial need of a broiler? Yeah. And maybe this kind of principle can help us in developing such a test. So we think that high density is perceived as something negative. And why is that? Because the animal doesn't like to be so close to another animal. That is perceived as negative. A little bit similar to a man approaching a woman and a woman liking to keep distance if she doesn't like the man that much. So you want to keep a personal space around you, and that if that person invades your personal space, then you create distance. Could a similar principle work in a behavioral test of the spatial needs of a broiler? So this is the principle. Chickens are social animals. So that means that if they have plenty of space, they will form groups. They will have a clustered distribution. However, if you limit the amount of space that this group of broilers has so severely, it will turn out that they will spread evenly in order to keep maximal distance between each other and preserve as much personal space as is possible under these very restrictive circumstances. And maybe the point where the distribution of the animal changes from heterogeneous or clustered to homogeneous is maybe a strong indicator that saying that this is the point where we think our stocking density is far too high. So we did a test to see whether that worked. So we again kept broilers in increasing stocking densities, going from 8 to 72 animals per pen, and these pens were 3.3 square meters. And we looked at the nearest neighbor distance, so how close they were to the closest other animal in the group. And we predicted that at very low stocking density, they would cluster together. So they would be closer to each other than predicted by chance. So a random distribution, which is the chance distribution, is indicated in the orange, and the observed distribution is indicated in the black. So as predicted, if the stocking density is very low, they will be closer to each other than predicted by random. But you can see, especially on week 5 and week 6, that already at 19 animals in the pen, that changed completely, where the axes were actually further from each other than predicted by chance. And we even came to the same conclusion when we did not a truly random distribution of the animals, but a simulated distribution of the animals, which takes into account that we know that the animals are attracted to walls, to places where they can eat in the pen, that there are places in the pen that are attractive or more attractive, and therefore they tend to cluster around these facilities. So even if we take account for that, we see that that their distance that they're keeping in the high density pens is further away than simulated or random. And the change happens when they go from 8 to 19 animals in the pen, which corresponds to 15 kilograms per square meter, which is much, much lower than what is maximally allowed in the EU as stocking density. Of course, these were tests done in an experimental setting with small pens, which are difficult to extrapolate to commercial conditions. But of course, measuring nearest neighbor distances is very time consuming, actually, to do it manually. So can the same principle be used in a more kind of convenient and more feasible test that can be applied on commercial farms? With the idea or the hypothesis being that if animals feel restricted in the amount of space that they have, would they not fill open spaces more quickly? So that's exactly the research question we did in this experiment. We asked the question, how quickly will chickens fill an empty space when they kept at low density versus a higher density? And so we changed these densities by keeping in the same size pens, but with different group sizes, one group of nearly 1,000 animals and then a group of 1,500 animals. So what did we do? We put down kind of 1 square meter frames on the litter, on the floor, and we let the animals habituate to that test, to that new object in the barn. And then on the day of the test, what did we do is we came in, we counted the number of animals that were in the square, we gently removed the animals from the square, and then watched how quickly that emptied space would be refilled again by the broilers, with the hypothesis being if the stocking density is high, this kind of refillment would be faster than if the stocking density is low. And we compare for the effect of the fact that in the high stocking density there are more animals by correlating it to how many animals there were in the square before the test took place, before we removed them. So we came up with this formula to see how quickly they filled in the emptied space compared to how many animals there were before that. And as predicted, we found a significant difference in which the filling ratio was higher in the high density group compared to the low density group. So maybe that type of test could be a better indicator of the spatial needs of broilers than simply looking at the stocking density. Let's turn now to an example for laying hens. And I said in the beginning, for me, animal welfare has mainly to do with the emotions and the feelings. So a very relevant criterion here is positive emotional state, for which in the laying hen protocol, 2 tests are being suggested, the novel object test and the QBA, and the QBA stands for qualitative behavior assessment. So just to explain the test, the novel object test is a very simple test in which an object which the animal doesn't know, has never seen before. Here it is suggested to have a novel object like this, of a stick with different color tapes around it, which is put in the home pen of the animal, on the litter in case of a non-cage system, or in the feeding trough in the case of enriched cages. And then you simply watch over time about how many animals come close to the novel object or even interact with it. The QBA is a test that is rather disputed in animal welfare science, with some people who think it's a very valuable test and other people think it's very subjective. What you do is that you simply observe in different time slots and different viewpoints groups of animals, and when you've done all your observations of different observation points, you score very subjective terms on a continuous scale from minimum to maximum, whether you think the animals are tense, content, calm, depressed, confident, and so on. So it feels very subjective to fill that in that list, and that is the qualitative behavior assessment. Are there problems with the welfare indicators of welfare quality? Well, I already discussed a couple, but there's also some inconsistencies. Some indicators give information about this very moment in time when you're doing the assessment. So they give a snapshot and can't be considered robust indicators. So if I do that assessment in the weekend, I would get possibly a different outcome when I do this same assessment a couple of days later. That is the case, for example, for indicators of thermal comfort. Indicators such as panting and huddling, they are very dependent upon the exact conditions at the time when the assessment is being done. And that's why no such indicators included in the dairy cattle protocol, just because they said this is not a robust indicator. And a second problem I referred to in the previous slide is with the QBA, the qualitative behavior assessment, being very subjective. And being subjective opens the potential for a judgment bias, for the observer having their own expectations influencing their scoring. And whether that is a true problem or not was investigated in this experiment using, again, veterinary students from the Ghent University. During a practical, they were trained to apply the QBA scoring methodology, and for that they were given a video of laying hens in an aviary system, and they were asked to apply the QBA and to score these 20 terms on this subjectively. And then they were asked to score a second video clip, which actually was exactly the same video clip as the one they had scored before, but it was turned around and we played a little bit with the lighting and so the students didn't think it was exactly the same video clip. And each group scored each video once, but one group was told the first video was taken from a conventional aviary farm, whereas that group was said the second video was taken from an organic farm, and the other group was given exactly the opposite information. And now we wanted to see whether the background information of where the video was taken, a conventional farm or an organic farm, could influence the outcome of the scoring. And before we did that, we also questioned them about how they thought the welfare of laying hens was on organic farms compared to on conventional farms. So we could relate on the individual level of the student whether they how they thought about organic farming, whether that had a bigger impact on the difference in their scoring between the two videos. And here you see the outcome of the statistical analysis, which was a principal component analysis. So what you see in the red lines are each of these 20 terms that the students scored when they had observed both video clips. In the dark circle, you find the scores for the conventional farm, and in the gray, you see that it shifted its position when they were given the false information that that video clip was made from an organic farm. And the shift that you see is towards the more positive terms, towards a more positive affective state. So terms such as whether the animals were relaxed, friendly, content, happy, confident, they were given higher scores, whereas negative terms such as depressed and frustrated were given lower scores, whereas in reality it was exactly the same video clip. We even found that the size of the effect, so the difference between the black circle and the gray circle, was directly related to the opinion of the students about organic farming. And so the difference would be bigger if they thought that the welfare of the hens on organic farms was better than on conventional farms. So this illustrates that really what should be done and what is good practice in science is that the observer should know whether the video recording was made on a conventional farm or an organic farm. But of course that is very difficult to do if you have to go to a farm and do a welfare assessment. You are influenced by factors you encounter there, and you can't be blinded to do the assessment. The next question I come to is we've covered what are essential requirements for a good welfare indicator. The second question is, what is a good combination of welfare indicators? But one of the things we want to exclude, for example, is that we have indicators that measure more or less the same thing, because then we are doing double work. So we preferably have indicators that are mutually exclusive, that measure different things. There's also the discussion depending on the application, do you want to have an as complete welfare assessment as possible, or do you want to focus on doing as many farms as many times as possible at the expense of being complete, but focusing on only the most relevant welfare aspects? And these type of decisions, of course, about which indicators that do I use, how do I combine them, they have to match with your definition of animal welfare. So it would be very strange for me saying animal welfare is to do with the feelings of the animals and me only using indicators of the physical health of the animal. That wouldn't match with my definition of welfare. The problem is that if we do say that we really want animal-based welfare indicators that also have information about the positive welfare of the animal, not only the negative welfare, then we have to admit that our field certainly at the moment is suffering from a shortage of good, validated, reliable welfare indicators. And that is a problem that possibly some aspects of welfare are much better documented by our current set of indicators than other aspects of welfare, and that not all dimensions of welfare are equally well covered by the indicators that we have. I will already talked about this slide, what the effect of that could be. A lot of the indicators that I use relate to the health and the biological functioning of the animals, whereas that is exactly what the very intense housing systems focus on preventing. They don't want any physical damage of the animal, but at the same time, these intensive systems, they offer far fewer opportunities for positive welfare and behavioral expression and positive emotions. But we have far fewer indicators of that. What is the effect of that is that our assessment tends to be biased in favor of the intensive rather than the extensive housing systems, just by our choice of indicators that we use. The ideal world, of course, would be where there was only one indicator that we could use that assesses overall welfare of the animals. Then we would only need to measure one aspect. Do they exist, what they are called iceberg indicators? Well, some have been suggested, but it's very much open to discussion whether with these indicators all aspects of welfare are truly covered. Iceberg indicators that have been suggested include, for example, the mortality of the animal, or the corticosterone built into the feathers or the scales of the animals, which give footprint almost of a whole history of being exposed to stressful events. Also, what we discussed before, the qualitative behavior assessment has been proposed as an iceberg indicator. And one that is still under scientific scrutiny and being discussed a lot is fluctuating asymmetry. And I will explain a little bit more about fluctuating asymmetry. So what is that? Fluctuating asymmetry is about small, random deviations from perfect symmetrical development of the body in those morphological features that are genetically programmed to be perfectly symmetrical. And if that perfect symmetry is not achieved during the growth, so if the left-hand side of the body differs from the right-hand side, that could be a reflection of how well the animal was coping with stress during its development. And if that is correct, it seems logical that fluctuating asymmetry could be an indicator of the animal's welfare, at least during the stage when the body is growing, so during its development. In order to test the validity of fluctuating asymmetry as a welfare indicator, we did a test where we went to 7 conventional farms and 7 organic farms, and from each of these farms we bought 10 random chickens and brought them to our laboratory and measured fluctuating asymmetry. So we measured the difference of various morphological characteristics on the right-hand side and the left-hand side of the body, and only focusing on those characteristics that were meant to be genetically symmetrical. But first we wanted to check whether it's truly the case that the welfare of the chickens from the organic farms was better than from those from the conventional farms. So we used again this set of commonly used welfare indicators for broilers, and again the tonic immobility, the latency to lie, the foot pad, hog burns, and foot pad dermatitis and breast blisters. And we used an index that was based on these welfare indicators, and indeed, as we had kind of hypothesized, the welfare index was better for the organic than the conventional broilers. And now fluctuating asymmetry. Well, yes, according to the hypothesis, the ones from the conventional farms were less symmetric as compared to those from the organic farms, exactly in line with the hypothesis. We did a similar type of study in meat rabbits to see whether the effect of housing also affects the amount of asymmetry. So now we compared rabbits that were housed in 2 different systems, one welfare friendly and one much less welfare friendly. So in the welfare friendly parts, they had enrichment as a platform, a shelter area, and a stick that they could gnaw on. And they were housed there in a lower stocking density with only 17 animals in the pen. Whereas in the more stressful housing conditions, there was no enrichment and there was a high stocking density of double the number of animals in the pen. Again, in line with the prediction, the amount of fluctuating asymmetry was lower in the animal-friendly pens compared to the barren pens. Here's a third experiment we did. We stressed half of the animals during the second week of life, and the stressor this time was cold stress. And you can see that the amount of fluctuating asymmetry was higher in the stress group compared to the control group. And what we found very interesting is that even when the animals were ready to be slaughtered, when they were 6 weeks old, the difference still was visible. So a stressor early in life, occurring at week 2, was not compensated during its further growth and was still visible at slaughter. This could indicate that measuring fluctuating asymmetry at slaughter could give information about the welfare during the entire growing phase of the animal. What's the downside of fluctuating asymmetry? It's very, very labor intensive. Doing all these measurements very precisely, measuring the left-hand side and then the right-hand side several times, it takes an awful long time. And you can even do two-dimensional measurements. You can look at the shape or the weight of bones. You can do even three-dimensional measurements and see how the shapes correspond and overlap between left and right. So it's very, very labor intensive. So unless we find ways of automating these measurements, it is not something that is very feasible for large-scale applications. So this was the last slide about the animal welfare indicators.